Hi, I'm Mark Parker. Welcome to my talk. I'm going to be telling you about otoacoustic emissions and how they could be used to identify hidden hearing loss in your patients. I am the Director of Audiology at St. Elizabeth's Medical Center in Boston, and I am an Assistant Professor of Otolaryngology at Tufts University School of Medicine. I have no conflicts, financial or otherwise, to disclose for this topic. It's a big goose egg. Um, I like to think of sensory neural hearing loss as a biological phenomenon. So we all know that we don't hear with our pinna, but the interesting or the fun stuff happens in the cochlea, in the inner ear. And the cochlea is composed of many different cell types. It's encased in bone. In the middle of it, the medulus, there's a large tunnel. This is a human cochlea. There's a tunnel in it called the medulus, but this is where the nerves run that connect the cochlea to the brain. And in the middle part of these tubes is the scala media. This is where the hair cells reside. And the spiral ganglia are here, and they're bipolar neurons when they have an axon that goes centrally to the brain to connect to the brain, and then peripherally to connect to the hair cells. But there's many different types of cell types, like I said, in the cochlea. There's inner and outer hair cells, typically one row of inner hair cells and 3 rows of outer hair cells. But there's also cells, fibrocytes, and ligaments that compose the lateral wall, the stria vascularis, and support many different supporting cells in the cochlea itself. So the hair cells sit on different supporting cells, like the Diter cells, for instance, or inner border cells or interphalangeal cells. And this is the cochlea of a mouse. Now, say you give the mouse a blast the sound of around 4,000 hertz. There's going to be damage in the corresponding region along the basilar membrane that corresponds to, say, 4,000 hertz. In this case, you see there's lots of damage. This stria vascularis is damaged. It's pulled away from the bone. And in this place here, there's actually no hair cells and just supporting cells left. But if you go farther away from, I say the ground zero region of damage, there could be variable damage. So there could be, in this case, there's an outer hair cell present and even one, inner hair cell present and even one outer hair cell present. And there's many supporting cells that are still present. So, the site of damage is interesting because it's not uniform along the length of the organ of Corti. Like I said, there's what I call ground zero, a place that could just be flat epithelium and really no cells present around the site of damage, the site of lesion. But the farther you go, either basally or apically, it would change. So there could be partial damage, and in some regions they'll be perfectly normal. So, say in the low frequencies, it's pretty common to have normal low frequency hearing, and then damage in the higher frequencies. So the damage is different along the length of the cochlea. In a previous paper, we spelled out 3 different forms of otopathologies that aren't easily detected in the audiogram. Specifically, synaptopathy, outer hair cell dysfunction, and then a third one called cochlear untuning. Now, this first 2 categories happen in people who have hearing within normal limits. So this is somebody coming into your clinic and maybe they're saying, I have a hard time hearing in background noise or some other hearing difficulties. And if you test their hearing on a normal audiogram, we would say, your hearing's normal, and then you kind of shoot them on their way. But we know now that there could actually be 2 different types of auditory dysfunction, otopathologies, that aren't being picked up in the standard audiogram. The first one is cochlear synaptopathy. And this has to do with the connections between the spiral ganglion neurons and the hair cells, specifically the inner hair cells. Normally, one inner hair cell connects to 10 to 20 spiral ganglion neurons, but we know that in some cases of temporary threshold shift, that the synapse between the spiral ganglion neuron and the hair cell could be damaged and they could be paired back. So this is called synaptopathy, and this happens in people usually with hearing between 0 and 15 dB HL. From 15 dB HL to 25 dB HL on average. A second type of otopathology is outer hair cell dysfunction, and this is like what I showed you with those earlier examples. Outer hair cells are more susceptible to damage than the inner hair cells, and so you can have a variable sites of damage. So even though someone's got normal, quote unquote, normal hearing or hearing within normal limits, we can measure outer hair cell dysfunction, and I'll show you that in a couple of slides here. The third type of otopathology is called untuning, and basically what happens is when there's too much damage, the damage of the outer hair cells is too suffice. The whole structure of the organochordy collapses. So basically, the tectoral membrane kind of falls down on the inner hair cells, and then there's a secondary untuning, a secondary effect on the eighth nerve, on the nerve fibers. So untuning basically is when the structure of the cochlea is so damaged that then it starts affecting auditory nerve function. And this happens in people with mild to moderate and greater sensorineural hearing loss. If you want some details, this paper describes it pretty well. So there is some debate among researchers on the causes and prevalence of hidden hearing loss. I'll define hidden hearing loss as an otopathology which is undetected by the standard audiogram. And there's two types of hidden hearing loss in persons hearing within normal limits. Like I said, there's cochlear synaptopathy. There's some debate whether it exists in humans, and if it does, what are the clinical effects? Some of these debate is really, I think, centered around selection bias and what, are we biasing our sample basically? My data suggests that cochlear synaptopathy actually happens, and it's relatively common in about 30% of persons coming in my clinic, but it doesn't play a significant role in hearing and noise or really in anything else I can measure other than that it happens. The second type is outer hair cell dysfunction. It's highly correlated with threshold changes in hearing and noise performance. This is the earliest damage. It's probably the best target for pharmacological and gene therapy or other types. So other types of treatments for biological treatments for hair cell regeneration, really. And then cochlear untuning happens in mild and greater cases as sensorineural hearing loss. So if you look at the standard audiogram, there's a range of normal that most people use, and that's with less than 25 dB HL, and then mild, moderate, moderate, severe. There's another category called minimal or slight sensorineural hearing loss. It's typically only used in pediatrics, but I would argue that you could use it in adults as well. So when you look at the otopathologies, we find synaptopathy can happen in people with thresholds less than 15 dB HL. But outer hair cell dysfunction typically starts to be significant in people with this mild or slight sensorineural hearing loss category. And then cochlear untuning happens in mild and moderate or mild and greater sensorineural hearing loss. So if we think about what's happening with outer hair cells, they're variably described as cochlear amplifiers or frequency filters or frequency tuning. We know that there's motor proteins in the outer, in the lateral wall of the hair cells, and that lets them elongate and contract to basically change the response to the inner hair cells. Now, when there's dysfunction to the outer hair cells, this affects the way that the inner hair cell responds. Actually, it helps hearing and threshold. It helps you, the outer hair cells help you hear in noise and help you hear at threshold. The threshold and outer hair cell function are correlated. So if you think of a lot of the data, some of the basic data comes from earlier studies in 60s, 70s, 1960s and 70s, where we know, like in a dead animal or a cadaver, that say there's a $4,000 stimulus, 4,000 hertz stimulus. I wish $4,000. And then along the basilar membrane, there's going to be a peak um, deflection at say 4,000 hertz. In a live animal, though, the peak is sharper. So, and we know that the reason for the sharpness is the outer hair cell tuning, the fine tuning. When the outer hair cells die, then you don't have this sharp tuning. And here's say here in black is the nerve fiber that's tuned, that's connected to this say 4,000 hertz stimuli. And what happens when there's untuning is that the tuning curve becomes less sharp and broader. So when I'm thinking about outer hair cell function, if you think about the whole length of the cochlea, it's a bank of frequency filters. And if there's some parts that are damaged, that's going to affect some of the frequency filters. And, it could even be all damaged in, say, someone who's profoundly deaf. So for those of you who don't use OEEs often, I'm going to show you how we measure our distortion product otoacoustic emissions. This is, say we're looking at a 2000 hertz tone here. This is the amplitude, and here's the noise floor in dark purple, and here's L2, so our stimulus, and we look at our F2, our F2 here. Here's our stimulus for say 2000 hertz, and then we measure the amplitude of the distortion product here. So, and this graphical readout gives us the amplitude of the distortion product in dB. And really, what this value actually, the way we have ours set up is we'll measure the distortion product, and it also measures the noise floor at an adjacent noise bin. And there's a couple of values that it gives you. You can either look at the distortion, the dB, the amplitude of the distortion product itself, or the noise floor. But really, the key here is look at SNR, the difference between these two, the different dB SNR. It's the difference between the noise floor and the amplitude. And this is what we look at. This dB SNR is the unit we use when we're analyzing outer hair cell function. So standard practice for DPOAEs places artificial limits on emission amplitude or robustness. And these are really generated or determined by the individual clinic. And they could either be, or they're commonly, either the 99th percentile of the emission or two standard deviations above the noise floor, with a minimum requirement of at least 5 dB SPL or 6 dB SPL or greater above the noise floor. So these are all artificial limits that people generally use when they're analyzing otoacoustic emission results. And then it's a binary choice. It's either present or absent. So here's the generated, absent, like accepted, which is present, or absent. So it's, basically it's either here or there. But what does this mean about the values in the middle? Some of them are present, some are absent. How do you interpret this? The data I'm going to show you uses dBSNR. We don't use any of these arbitrary limits, so we just look at dBSNR, and I'll show you how you could use this in your clinic. So there was a previous paper in 1997 from Gorga et al., and they had a large subject, 806 subjects. And they were basically looking at, or we're looking at what they were looking at too. Probably the best thing to look at is here, like normal, mild, moderate. So they measured OEEs, these dBSNRs, and what they showed is that people even within normal limits, normal hearing less than 25 dBHL, they're like the lower their thresholds, the better, or the higher the dBSNR was, and it decreased to a certain level. And we've been doing similar research. And sorry, I passed that slide too fast. We're doing similar research, and we find similar results. In this is what we're finding. If you plot the dBSNR of the emission by their pure tone average, or their frequency actually, we see there's a difference when people who've got, like, well, I'm going to redefine normal. People who've got thresholds of less than 15 dBHL. So if you look at people with thresholds of less than 15 dBHL, they've got a nice robust emission. But even people with minimal or slight sensory neural hearing loss have a significantly decrease in their dBSNR values. And then, it gets worse with mild or moderate sensory neural hearing loss. And if we could plot it out by another way, by looking at the pure tone average and looking at the dBSNRs of their emissions, and then we just broke everything out in color here. So people who've got normal, which is PTAs of less than 15, have higher or more robust dB, like outer hair cell function. When you get to minimal sensory neural hearing loss, it's decreased from there, and then mild to moderate, it even goes down. It's a very linear function with a high R. It's a high correlation. So basically what this is showing is that threshold is highly correlated with emission amplitude or robustness. And then what we looked at was, okay, great. So it looks like outer hair cell function is tied to threshold, but is it also it's correlated with threshold? Is it also correlated with hearing and noise performance? And the answer is yes. So here's a different DPOE SNRs at different frequencies, where we're looking, I think the N of this was 84 too. And then we did different speech and noise testing, 3 different speech and noise tests. This is time compressed word rec in time, 10% reverberation and 45% time compression. This is word recognition scores in 0 dB SNL's speech weighted noise, and here's the quick SIN. So I'm just going to focus on the quick SIN, but the story is the same with all of them, in that speech and noise scores are highly correlated with outer hair cell function. So the better the outer hair cell function, the better the speech and noise score is. And this shows the same data. So here's just looking at the quick SIN, and in the quick SIN, the lower score is the better performance in noise. So people in this normal group, new normal group, is less than 15 dBHL, has significantly better or lower scores on the quick SIN than people with minimal sensory neural hearing loss or mild to moderate sensory neural hearing loss. And we could also break this down in different groups, where here's a normal group, and it's a linear function, and this red is the mean dBSNR score for this group, which is in this case of like 20, just at 3,000 hertz. And then the quicksin, the means quicksin score in this group is at 1. And then keeping everything the same, just moving, adding the minimal group, we could see that there's a decrease in the group mean. And then when you get to the mild and moderate scores, there's no more correlation between hearing and noise performance and outer hair cell function. So in English, what we're showing is that people with better hearing, better thresholds, perform better in hearing and noise, and it starts to decrease even in someone with 15 to 25 dB HL thresholds. So this is someone with quote unquote has got normal hearing. So somebody comes into your clinics with quote unquote normal hearing, they could be experiencing, and if they're saying, oh, I'm having a hard time hearing a noise, they could be experiencing a hard time hearing a noise because their outer hair cell functions could be compromised. So I'm going to show you how to use this in your clinic. You could use both quicksin or DPOE SNRs, but we need a larger N right now, and we're working on it for clinical relevance. So basically, here's the normative values at different frequencies. 1,000. So this is the FT frequency of 1,000, 1,500, up to 8. I'll say 1,000 and 8,000 could be problematic just because the errors are higher there. The meat of it is between 15,000 and 6,000. And the numbers in green here are the 95% upper confidence levels for the means. So in English, what this means is that a value below these numbers here shows suggests that there's outer hair cell dysfunction, not the presence or absence, but there's dysfunction happening. So anything greater than 10, it's normal outer hair cell function. Anything lower than 12.9 here is showing outer hair cell dysfunction. So we're doing sensitivity and specificity measurements here to see if we could predict outer hair cell hearing and noise difficulty using outer hair cell function or dysfunction. So the methods that we're using is we've recruited 50 persons hearing within normal limits. We ask them to self-rate their difficulty hearing and noise, and then really that's what that's our metric we're using, our clinical metric. But we also added bothersome tinnitus or bothersome hyperacusis. There's no evidence linking tinnitus or hyperacusis to outer hair cell dysfunction. So these are our negative controls. And then we measure the DPOE, DBSNR, like I described. Diminished responses was defined as the DBSNR less than the up or down, that 95% CI that I mentioned. The end of this is pretty high. We excluded 1,000 and 8,000 hertz from the analysis, and if there's a difference between ear, we just looked at the worst ear. And we said those persons exhibiting a DBSNR above these values is normal and below these values is abnormal, so values in green. And then we calculated sensitivity and specificity in as far as OAE function as far as predicting self-reported hearing and noise difficulty, tinnitus, and hyperacusis. So we'll get to this. So here's a case study first. This is a person who came in. He was 35 years old, late 30s. He was a teacher, and he said that he was having a hard time hearing in his classroom. So here is his hearing test. And again, he's got hearing less than 25 dB HL. And we looked at his speech testing, and his speech and quiet looked fine. And then we did, so if somebody's coming into your clinic and saying that they're having a hard time hearing and their audiogram looks normal, what do people do? Since we're studying this, I did the QuickSyn on them. And on the QuickSyn, according to our data that I showed you earlier, a normal QuickSyn score is 1 or less. The QuickSyn score is SNR loss. So a normal, according to the manufacturer, it's 3 or less. Our data, but the manufacturer didn't know about our outer hair cell dysfunction. So our data shows that outer hair cell dysfunction starts at a number greater than 2, an SNR loss greater than 2. And this person had an SNR loss of 17, which was severe, basically a severe difficulty hearing and noise. So how can you explain this? The guy's got normal thresholds, normal speech and quiet, but he's got horrible speech and noise. And then we did OAEs on this person. And so again, some of these are accepted and some of these were rejected. So how can you interpret that? It's either presence or absence. If you add in addition to the presence or absence, our criteria, you can see that he's got lots of, even in some of his accepted scores, they're diminished. So there's a lot of diminished amplitudes. So to say this in English, person comes in reporting difficulty hearing and noise. He's got quote unquote thresholds within normal limits on a standard audiogram, airborne and speech. But if you do speech and noise testing, you may be able to find a significant problem. And if you do OAEs, you could also find a different problem if you redefine what your criteria for normal or abnormal is. So instead of saying presence or absence, you could say diminished or robust. And in this case, he's certainly in his left ear, he had diminished outer hair cell function. So when we plug, that was his case study. Now, if we look at the 50 people that we enrolled in the study, 50 people with normal hearing, and we measured the sensitivity and specificity. What we found was that the sensitivity of using OAEs to predict the OAE SNRs to predict difficulty hearing noise using the quicksand, what we found this test was 80% sensitive and 71% specific, with a PPV 57, a negative predictive value of 88%, and an accuracy of 74%. So how do these numbers fit in with anything else we do in audiology? If you look at ECOG to measure Meniere's disease, the sensitivity and specificity is low. It's only 60% sensitive in diagnosing Meniere's disease. It's got a high specificity though. In contrast, probably the highest sensitivity in any of the tests that we use is caloric irrigations to diagnose unilateral weakness, where we get 90% or greater. Also ASSRs for pure tone screening, and the specificity is high. So our results are more in this round here, the 71. So more in the realm of 70, so 70% and 80. So we're looking at about here tone burst ABRs to diagnose pure tone screenings. So again, in English, it looks like we're in the process of publishing this, but it looks like OAEs can be used to diagnose hearing and noise difficulty with a sensitivity and specificity that is commonly accepted in our field. Now, for the negative controls for the bothersome tinnitus, there's really the sensitivity and specificity were very low, and same for hyperacusis. So basically, the conclusions that we have based on this study are that OAE, DBS, and Rs have the highest sensitivity and specificity, positive predictive value, negative predictive value, and accuracy in correctly identifying hearing and noise difficulty. They have relatively low ability to correctly identify tinnitus or hyperacusis. Those were our negative controls anyway. But these functions, these measurements may be suited for screening purposes for hearing and noise performance and for hidden hearing loss. I think, yeah. So what does that mean? Does this mean that we could use it in any other way, OAEs, for anything else? And particularly, what about hearing aids, right? So typically we fit people with hearing aids who've got mild or moderate sensorineural hearing loss. But if we can measure an otopathology in people with minimal or slight sensorineural hearing loss, and these people also are demonstrating that they have a problem hearing and noise, could we use hearing aids, particularly with wide dynamic compression and adaptive microphone adaptive directionality, noise reduction functions of the hearing aids? Would they be beneficial in people, I guess, off-label? And we have a little pilot study with just an N of 20 where we fit people with hearing, people reporting, problems, or actually, sorry, this is a hearing aided performance in one of our hearing and noise tests. And what we see is, after a month after fitting with hearing aids, people do better. Right? This is people with within normal limits, mild and moderate hearing loss. But if you break this down into people who've got hearing, within less than 25 dB HL, so hearing within normal limits, versus people with milder or greater hearing loss, what we see in both cases, the aided group does better. Even if you're hearing within normal limits, if you put a hearing aid on, you can do better. So it does suggest that this group here might benefit from hearing aids. So, the normal, so here's a normal standard that people use, and there's a potential standard of including minimal in this category of hearing loss. because we could detect an otopathology that also has a clinical correlation. And right now, here's a current hearing aid fitting range, but if we could measure a pathology with a clinical correlation that can be improved with hearing aids, then we could potentially expand our fitting range and help our patients. So with that, I would just like to acknowledge some of the students that are helping me with this project now: Kelly Stahura, Christian Burden, Kate Bergen, Sophia Pesner, who works with me at St. All right, thank you for listening to my talk.